Hi, good morning or good afternoon. My name is Enliang Li, and it's my great pleasure to present the paper number 268, our paper, Improved GPU Implementations of the Paired chain memory algorithm for DNA sequence alignment. And I want to thank to all authors on this slide for their contribution, and I'm grateful for ICCD committee for giving me this opportunity to present this work. So the first part, we want to cover the section 1 and the 2 in our paper. Just want to give you some background and a motivation of why we are looking into this work. So the first terminology we mentioned in our paper is the next generation sequencing. So the NGX technology has the feature of low cost and high performance to their previous counterpart. And the thing that we can imagine is that will become more demanding on computational power. And there are two sources for more computational power. The first one is the better fabrication technology that will be innovated hardware like GPU and better FPGA. And the second part is the improved algorithms that could be fit inside this better hardware. And the GATK refer to genome analyzed toolkit that will appear a lot of time in this paper as well. And this is open source and developed by the Broad Institute, and it's a very popular and widely used toolkit for DNA analysis. So we want to take a look at why people are focusing on ParaChain and why we want to accelerate it, because it could be easily taking 70% of the total execution time of the GATK heptide caller on a portion of the NA12878 sample. We have reference for that actual data. So this is just give you a sense of like how much time and the majority of the time will be used for ParaChain for algorithm. And that could be different across different samples. So in next sections, we want to cover the section 3 in our paper. So here for GPU related works, we refer to the paper reference 3 and 4. And the FPGA related works refer to paper reference 5, 6, and 7. For GPU previous work, there's two separate methodology proposed, and they're implemented in the separate implementations. The first one is intertask, and the other one is intratask. The intertask is kind of like you want to parallel compute different pairs of sequences alignment together. And the intratask is actually going inside each pair of the sequence alignment and trying to explore the parallelization during the for algorithm update or execution. And for FPGA work, the paper reference 5, 6, they're using something called a processing element structure. Actually, you can take it as the FPGA version of the intratask parallelization implementations. And paper reference 7 is actually using posted number format to represent a floating point. So actually, they will need to implement the corresponding like the operations for that posted number format support. So our observation here is that previous talk do not discuss too much on memory transfer overhead and how to mitigate it. And also they assume the transition and emission probability matrices. These two are very important for calculating the total likelihood are done on the CPU. And previous work does not provide good scalability. We have better scalability in terms of that we can adjust the length of the processing unit. And also our data structure is more friendly to GPU. So this part we're going to cover section 4. So the first terminology of the software concept is the processing unit. So processing unit in our algorithm is composing of multiple GPU threads, and they need to have the same shared memory access. So they can exchange the data through shared memory that will be discussed in a later slide. And the length of the processing unit is actually decided by two factors. The first thing is that how large is the current workload in terms of the read and the halotype lengths of the DNA sequences, and also the warp size of the current GPU that the algorithm is running on. So here the computational parallelizations The across-state parallelization is actually meaning the match, insertion, deletion referred to the different color here. They're independent and could be organized to run together. But this requires us to have a way to identify which state each thread is working on. And for the cross-diagonal parallelizations, the figure on the left here is assuming that the processing unit have a length of 4. So the yellow one is currently working on, and the green one is what it will work on next. And if the bottom left one is the leading for currents calculating cells, the moving direction is indicated by the red arrow. So you will first go down, and then it will first go horizontally to the right until we update the last, the bottom right cell for the total likelihood. So the memory strategy here is also covered 2 parts. The first thing is that the shared memory strategy. So we can see we don't actually interested in every cells in the forward matrix. So we only care about the total likelihood at the bottom right. So we don't need to keep track of the whole forward matrix. Instead, for the green cells that are updating the current anti-diagonal, we only need to keep track of 2 direct yellow rows that in front of it. So here, 1, 2, 3, these cells are the necessary one to calculate the green one. And the other part is that we can using the multiple CUDA stream to kick off the host-to-device transfer for necessary data when we are doing the initialization or computations. So this part, we are seeing a clip here as 320. So actually, we are having 2 different kernel size. So to make sure that only necessary resources are allocated for the workload. So this part, we're going to cover the section 5. So here, the top left figure here is that 10-second data set, referred as 16. And the one here is that the different methodology that's covering using the real GATK workflow and a speed up of 783 to the Java baseline, 31.88 to the previous C++, and 1.5 to the state-of-the-art GPU implementation on the same devices. Okay, thank you so much. And this is our contact information. And this work is based upon a work supported by National Science Foundations under 2 different grants. Thank you.